Dear students, our lecture today is about hypersensitivity reaction, part 1. Here are some key definitions will be mentioned during the lectures. Learning objectives of the lecture are hypersensitivity. How does it differ from normal immune response? Classification of hypersensitivity reaction. Type 1 hypersensitivity, immediate type, or we called IgE-mediated mast cell degranulation. Type 2 hypersensitivity, antibody-mediated cytotoxicity. To recap, appropriate immune responses detect foreign antigen. Sufficient number of antigen-specific lymphocytes are generated by clonal proliferation. Appropriate class of antibodies produced to clear the infection by binding to the surface of the pathogen. Formation of IgM or IgG containing immune complexes triggers the activation of classical complement pathway. The IgG and complement component opsonize the pathogen for subsequent phagocytosis. Interferon gamma, macrophage activating factor, is produced by NK cell, T helper 1, and cytotoxic T cells as a response to intracellular pathogen. Now, hypersensitivity, how does it differ from normal immune response? Immune response that extends beyond its usual boundary of recognizing only foreign pathogen to also encompass the innocent environmental antigen leading to tissue damage or immunopathology. The mechanisms underlying hypersensitivity reaction are the same as those normally employed by the body in combating infection. The problem is that they are occurring with much too high an intensity directed against antigen that pose no threat, taking place at locations in the body that are inappropriate. Now, hypersensitivity reactions can be classified, originally classified by John Combs into 4 types of reactions, and this classification remains broadly useful. Type 1, immediate type IgE, mediated mast cell degranulation. Type 2, antibody-dependent cytotoxicity. Type 3, immune complex mediated. Type 4, delayed type cell-mediated hypersensitivity reactions. Some subsequent additions to the original classification have included type 5, stimulatory hypersensitivity, and innate hypersensitivity. Most allergies, hay fever, asthma, eczema, and food allergies are mediated by type 1 hypersensitivity, although some forms of eczema are due to type 4 reactions. Also, in a particular disease, more than one type of hypersensitivity may be operating. Now, type 1 immediate type IgE-mediated mast cell degranulation. When first exposure to antigen, or we call the allergy, this antigen binds to dendritic cell. On dendritic cell, each dendritic cell will phagocytosis and degrade antigen into small peptide and present antigen peptide in combination with MHC2 naive for CD4 naive T cell. After encounter of CD4 naive T cell with this complex, it will activate and differentiate and secrete interleukin 4 differentiation into T helper 2. T helper 2 secretes interleukin 3, 4, 5, 13 to activate B cell and mast cell. Activation of B cells leading to B cell class switching for IgE synthesis. B cell then after that will secrete IgE. This IgE will bind with specific receptor on mast cell, and it has a high affinity to Fc epsilon R1 receptor on mast cell. In second exposure to the same antigen, to the same allergen, binding of antigen with IgE on mast cell. Now, why antigen bind with IgE? Because IgE made specifically to that antigen from the first exposure. Now, after binding with antigen with IgE on mast cell will be there will be a signal through its receptor Fc epsilon R1 inside the cell to increase the synthesis of its mediators inside the cell, and then leading to mast cell degranulation, releasing histamine and other mediators. Preformed mediators that release after degranulation of mast cells, such as histamine, heparin, tryptase, trimase, carboxypeptidase, eosinophil, neutrophil, and monocyte chemotactic factor, platelets activating factor, serotonin. Anaphylaxis. Anaphylaxis results from mast cell degranulation and releasing its mediator. It's characterized by intense constriction of bronchioles and constriction of smooth muscle, dilatation of capillaries. Now, atopic allergy. Clinically, type 1 hypersensitivity can manifest itself as immunological reaction in gastrointestinal tract, eczema, atopic dermatitis, asthma, and hay fever. Hay fever is seasonal conjunctivitis, rhinitis. These conditions often occur in the same individual and develop in an ordered sequence called allergy march. Now, what's the allergy march? Here in this diagram, infant and children with gastrointestinal and cutaneous allergy causing GI tract and eczema allergy have 2 to 3-fold increased risk of later developing asthma and hay fever after, or rhinitis in that case. This is the allergy march. Of atopic allergy. Genetic factor, genes encoding HLA, cytokines, etc. Various environmental factors, age, sex, infection history, nutritional status, allergen exposure, the overall ability to synthesize IgE. The higher the level of IgE in the blood, the greater is the likelihood of becoming atopic. Diagnosis by using skin prick test. Sensitivity normally assessed by the response to intradermal challenge with antigen, release of histamine and other mediators rapidly produces an immediate virulent flare within 30 minutes and then subsiding, followed by a late phase reaction in the injected skin lasts for 24 hours. Dense infiltration with eosinophil and T cells in the site of injection. Now, measurement of allergen-specific serum IgE. Correlation between skin prick test responses and serum IgE is fairly good. In some instances, intranasal challenge with allergy may provoke a response even when both of these tests are negative, probably as a result of local synthesis of IgE antibody. Several points in the chain of reaction provide target for therapy. We can have an idea of which targets we use the therapy against it. Allergen avoidance, desensitization, immunotherapy, blocking the action of IgE, inhibition of effector cells, mediator, antagonism, limiting chronic inflammation in that case. Type 2 hypersensitivity, antibody-dependent cytotoxicity. It occurs through two distinct cytotoxic mechanisms: complement-dependent cell-mediated cytotoxicity and antibody-mediated cellular cytotoxicity. Now, the complement-dependent cell-mediated cytotoxicity, when the target cell binds with antigen, antibody, just like IgG or IgM, bind with antigen specifically. This will lead activation of complement classical pathway through C1 to C4, C2 in order to activate and split C3 into C3b. C3b, as opsonine, will bind with target cell. In that case, phagocytic cells have specific receptors C3b, bind with this opsonine and leading to phagocytosis of target cell. or it can be done through the C5, 6, 7, 8, C9 to MAC formation, membrane attacked complex formation, and leading to cell lysis, the target cell lysis in that case. Antibody-dependent cellular cytotoxicity, when the target cell antigen on target cell binds specifically with the antibody, just like IgG or IgM, the Fc gamma R receptor on the antibody will bind with specific receptor on NK cell, neutrophil, monocyte, or eosinophil, in that case leading to target cell killing by these cells. Type 2 reactions between members of the same species, autoimmune reaction. Autoimmune reaction, just like antibodies to the A or B antigen on the ABO blood grouping, occur spontaneously when antigen is absent from the RBC surface. Thus, a person of blood group A will have anti-B, blood group B will have anti-A, and blood group O will have both anti-A and anti-B. These antibodies, called isoheme-agglutinin, are usually IgM and belong to the class of natural antibody. The natural antibodies are immunoglobulins present in the absence of exogenous antigen stimulation. It's naturally occurred in the body. On transfusion, mismatched RBC will coated by isoagglutinin, in that case causing severe complement-mediated intravascular hemolysis. Maternal antibody. In maternal antibody, the Rh versus blood groups. A mother with an Rhd negative blood group can readily be sensitized by RBCs from a baby carrying Rhd antigen, Rh positive, and form anti-D antibodies. This occurs at the first birth of the first child, when a placental bleed can release a large number of baby's RBCs into the mother. Anti-D antibodies are IgG. Why IgG? Because IgG can cross placenta, while IgM cannot, because it's pentameric. IgG then able to cross the placenta in any subsequent pregnancy to the fetus. Diagnosis by indirect COMS tests. During subsequent pregnancy, anti-D antibody will react with D antigen on the fetal RBC, leading to RBC's destruction through opsonic adherence, causing hemolytic disease of fetus and newborn HDN, erythroplastosis fetalis. In this disease, IgG from mother crosses the placenta, targeting the fetus RBCs for destruction. Examples for type 2 hypersensitivity diseases: neonatal autoimmune thrombocytopenia, and solid organ allograft. What do we mean by allograft? Transplant of an organ or tissue from one individual to another of the same species with a different genotype, not identical twin. Solid organ allograft in case of mismatched MSC, autoimmune type 2 hypersensitivity, autoimmune hemolytic anemia, it's autoimmune type, autoimmune hemolytic anemia. Let us discuss something about it. It's a rare red blood cell disorder, an immune disorder. IgG and IgM bind to antigen on transfused RBCs, targeting donor RBCs for destruction. Idiopathic thrombocytopenic purpura. It's an immune disorder in which the blood does not clot normally because the immune system destroys platelets, leading to decreased number of platelets in the blood. Also in Basrah syndrome, rare autoimmune disease in which antibody attack the basement membrane in lungs, kidneys, leading to bleeding from the lungs and kidney and leading to kidney failure. Also in myasthenia gravis. Myasthenia gravis is an autoimmune disease of the neuromuscular junction caused by antibodies that attacked component of the postsynaptic membrane, impair neuromuscular transmission, and lead to weakness and fatigue of skeletal muscle. Thank you very much.